The overall goal of the following experiment is to locate and quantify microRNA expression within kidney tissues. This is achieved by first hybridizing a deoxygenin-tagged probe to a given microRNA within the kidney tissue. As a second step, an antibody to deoxygenin, which has a conjugated alkaline phosphatase, is incubated with the tissue. Next, an NBT-BCIP substrate is added to the tissue in order to visualize the location of the microRNA through development of a blue-colored product at sites of the hybridized microRNA probes. Results are obtained that show where microRNAs are expressed in kidney regions and nephron segments based on in situ hybridization of microRNA complementary probes.
This method can help answer key questions in the field of kidney research, such as where microRNAs are expressed within the kidney. This provides us with information about the role of individual microRNAs in kidney function and regulation, as well as pathology.
This procedure uses formalin-fixed, paraffin-embedded kidney sections that are cut onto microscope slides at a thickness of 5 microns. To prepare the kidney sections for analysis, first remove the paraffin from the tissue by 3 washes in fresh xylene for 5 minutes each. Rehydrate the tissue sections by 5-minute incubations in decreasing concentrations.